Imagine a world teeming with life, a primordial soup of single-celled organisms. These tiny pioneers faced a constant battle for survival, surrounded by a myriad of threats. How did they develop the tools to defend themselves? Billions of years ago, the first life forms emerged as simple prokaryotic cells. These organisms faced a constant barrage of damage causing foreign proteins and toxins. Some of the toxins would alter the expression of the genes of such single-cell organisms. To survive, they developed basic defense mechanisms. Cell walls are rigid structures that surround the plasma membrane of prokaryotes. They are primarily composed of peptidoglycan, a complex polymer of sugars and amino acids. Cell walls provide structural support, maintaining the shape and integrity of the cell. They prevent osmotic lysis, which occurs when the cell absorbs too much water and bursts. Cell walls act as a physical barrier, preventing the entry of harmful substances. The cell wall acts as the first line of defense. Prokaryotes lack nucleus, mitochondria, and other organelles that characterize the advanced present-day cell. The cytoplasm within their cell membrane composed mainly of water that also contained enzymes, salts, ribosome, and single DNA within a nucleid, besides homologs of actin and tubulin proteins. The helically arranged building block of the flagellum, flagellin, is one of the most significant cytoskeletal proteins of bacteria. It provides structural backgrounds of chemotaxis, the basic cell physiological response of bacteria. These organisms direct their movements according to certain chemicals in their environment. This is important for bacteria to find food like glucose by swimming toward the highest concentration of food molecules, or to flee away from the toxins. Enzymes. They can break down harmful invading molecules, such as nucleic acid, or toxins, rendering them harmless. Prokaryotes sense the foreign materials through their receptors and mount a response either moving away or secreting enzymes to degrade them. Enzymes are also involved in repairing DNA damage caused by radiation or other harmful agents. Unlike eukaryotic cells, prokaryotes doesn't have phagocytosis. However, before ancient prokaryotic cells, before evolution of eukaryotes, it is believed that they used to have phagocytic mechanisms. Quorum sensing is a cell-to-cell communication system used by prokaryotes and other microorganisms to coordinate their behavior in response to changes in population density. This process involves the production, detection, and response to signaling molecules. These are called as autoinducers. Once after these autoinducers detected by interacting prokaryotic cells, the concentration of autoinducers at certain level leads to the activation of genes within them, this causing production of various molecules, including enzymes, proteins, and virulence factors. The other mechanism is such gene modification may lead to formation of defensive biofilm. Biofilms can provide protection against toxins and other environmental stresses. Quorum sensing can facilitate the exchange of genetic material between prokaryotes. It can lead to the acquisition of new traits,